All, I hope you are okay. Sorry for inconveniences caused. We came in and then we screwed out. So I just want to say sorry for that. There was a lot of rains and I couldn't be audible enough. So we just tuned in. We came in for very few minutes and then we left. So I want to say welcome. This is Chemistry Wednesday. Your presenter is Teacher Kirwa. We have done chemistry lessons all time since we started our lessons, and these online lessons back then in May. So we have done a lot of chemistry topics or chemistry tutorials. Today I want us to change our gears a bit and then we look at biology. So we are doing biology, we are doing cell physiology today, and for those who were around, I had done some introduction, although we are now doing it again. So we want to say thank you kwa ule ulikuwa umetuletea mvua. Asante kwa sababu otherwise karibuni sana wapendwa. Go to my YouTube channel Teacher Kirwa, red button, weka kidole, then you keep on viewing the videos that are there. So thank you, welcome, let us interact. Keep your comments coming and the likes there at the comment box. So cell physiology. Now cell physiology is the study of cell functions, cell structures and functions. Study of cell structures and function. In our previous lesson, in a previous lesson of this or a previous topic, you look at the cell and you say a cell is a basic functional unit of any living organism. Now a cell, many cells come together and then they form what is called a tissue. Many tissues work together to form an organelle, an organ. Many organs come together to form a system, and then several systems come together to form a human being. So that when you stand like this, you are made up of a breathing system, breathe. You are made up of a nervous system for coordination of responses in the body. We also have an excretory system that takes away the waste products, the indigestible waste products, and the materials that were not used during digestion, and all the waste products. We have several systems. We have the endocrine system, we have the digestive system. So all those systems make up an organism. Now the organism, the systems are made up of organs. For example, if you say breathing system, we have the nose, we have the lungs, we have the alveolus, okay, we have the trachea. Those are the bronchi. Those are the organs. Now those organs contain tissues, and then the tissues contain the cell. So a cell is basic. So it is basic and then it is functional. Now for a cell to function, the materials have to be taken in, raw materials have to be taken in, and after the raw materials are used to manufacture or to make products, then the products are taken out to various parts of the body where they effect their functions. So cells physiology is the study of cell structures and function. The most important part of the cell here of the cell is the cell membrane. This is what we said for those who had tuned in. By the way, thank you. Nilikuwa nimeona deputy wangu, nilikuwa nimeona classmate wangu, nimeona wanafunzi wangu. So thank you. I hope you are back again. If you aren't back yet, then you will view these videos later on. So the most important part of the cell organelle that we will talk about throughout this course, this topic, is the cell membrane. Now the cell membrane is made up of a phospholipid layer in between protein layers, so that we have a protein layer, and then we have the okay, it does not look exactly like this. I'm just using this to explain the layer, the function of in between. So we have a protein layer. So this one is a protein layer, this is a protein layer, this is a phospholipid layer. So the phospholipid layer is sandwiched between two protein layers. Now, so that means the protein layer is above and below. Now, so we say the cell membrane is said to be a lipoprotein. So it is lipoprotein made up of phospholipids and proteins. So properties of cell membrane. Properties of cell membrane. Properties or characteristics, you can say properties or characteristics of cell membrane. Now, characteristic number one is semi-permeability. Semi-permeability is the ability of the cell to of the cell membrane to allow small particles, substances of small particles, to pass through it. So we say it allows particles or molecules of small size through it, but doesn't allow particles of large sizes. So it allows particles of small size to pass through it and does not allow those of large size. So we, in short, the cell membrane is said to be selectively permeable. So the cell membrane is said to be selectively permeable. Selectively permeable is the same as semi-permeable. Now there are three types of membranes that we know, that we talk of. We have three types of membranes. I'm just mentioning this. We have membranes which are permeable. We have membranes which are semi-permeable. Okay, and then we have membranes which are impermeable. Some membranes are permeable. Permeable means it allows molecules of all types to pass through it, whether they are small, whether they are large, whether they are in between small and large. All those particles pass through. So it is permeable. So we say the membrane is permeable. Now some are semi-permeable, like the cell membrane now. The cell membrane is semi-permeable in the sense that it allows some substances to pass through it and does not allow others to pass through. It is selectively permeable. Some membranes are impermeable, or some substances we say they are impermeable. Impermeable or impervious or non-permeable, any of them can do. So when you say impermeable, it means it is completely impermeable. It does not allow molecules of any kind to pass through them. So these are the three types of membranes that we have in biology. Number one property of cell membrane is that it is semi-permeable. Number two is sensitive to changes in pH and temperature. It is sensitive to changes in temperature and pH. Just to review, we have mentioned that the cell membrane is made up of a protein layer. Now, essentially, when you look at proteins are enzymes in nature. They are enzymes in nature, and enzymes are sensitive to changes in pH and temperature. So we because it is made up of enzymes, we can say high temperatures denature enzymes. Denature or destroy or kill, any of that can do. So high temperatures denature enzymes, and low temperatures make enzymes inactive. Now, high temperatures denature enzymes. Now that is why the saizi wanapima temperature, yeah? Hii maneno ya COVID, if it is there. Oh, sorry. So wakipima temperature, the temperature should be 37. Okay? 37 degrees centigrade. This is the temperature of the normal human being. So ukipimwa temperature na wapate ni 40 and above, that is very dangerous, and then you are taken into quarantine or whatever, because it is not supposed to be above 40. If it goes above this, then likely the cells will die. If the cells die, you automatically also die. So that is it. Now, when the temperatures are below 37, the enzymes will become inactive. You know, when the temperatures are very low, sometimes even like now, when the temperatures are very low, it is very cold, you shiver, you become inactive. Okay, so similarly the enzymes become inactive at low temperatures, but at very high temperatures they are denatured or they are killed. Okay, number 3 property number 3 is that it possesses electric charges. A cell membrane, the cell membrane is charged. It has positive and negative charges, so it possesses electric charges. So that is the cell membrane. So next we look at physiological processes. Now, substances pass through the cell membrane via very many physiological processes, but we are going to discuss only 3 at our level. We are only discussing 3 at this level to enable you to continue with school. If we look at the others, most of you might leave school. So because you are at this level, this is our Form 1 student fresh from primary, tunaangalia tu kidogo. After all, this is what was done also in primary science. So the first one we look at is diffusion. So the first one is diffusion. Kila mtu anajua diffusion, eh? When you are seated in some place and then ugali is cooked, eh? Those of you who grew at those times, there was a time when you were in school, harufu ya ugali inaanza kutoka, inaanza kuingia, na ukianza kusikia harufu ya ugali unaimba ile wimbo ya kwaheri mwalimu, si ndio? You used to sing kwaheri mwalimu. Kwa nini? Umesikia harufu ya chakula. Hiyo harufu ya chakula ilikufikia aje? It is through diffusion. So it is through diffusion that you can smell piece of chapati, okay, where the fisi was alichanganyikiwa aende hapa ama hapa, akaanza kuenda hivi, okay. So you have you can feel that smell, you see, by the process of diffusion. Also, you know, when we were in school those times, there are teachers who used to like marashi and so on and so forth. So angepita kwa mtu akipita kwa dirisha, unasikia unajua huyo ni mtu fulani. Hiyo harufu ya marashi ilikufikiaje? It is through diffusion. So diffusion is the movement of particles or molecules. It is the movement of molecules from a region of high concentration to a region of low concentration. If you want to demonstrate diffusion, the best way to do it, you take a bottle of marashi, yeah. You take a bottle of marashi, go to one corner of the class, and then you open it, okay. When you open it, after a few minutes, everybody else in the classroom will be able to get that odor. How is it that possible? Because inside the bottle here, the molecules are highly concentrated, or the particles are highly concentrated here. Now when you let it out, the high concentration here, the environment around is said to be lowly concentrated. So when it is open, it moves very fast because it is moving from a region of high concentration to a region of low concentration. Factors affecting diffusion. Factors affecting diffusion. These are factors which either increases the rate or reduces the rate of diffusion. The first one is size of the molecules. Size of the molecules. The smaller the size of the molecule, the faster the rate of diffusion, because when a molecule is small in size, the movement is very fast. It is swift as opposed to large sized molecules. So the smaller the size of molecules, the higher the rate of diffusion, and vice versa. Number two, is what is called diffusion gradient. Diffusion gradient is the difference in concentration between two solutions. Ukisema high na low, that difference where you have high and low is diffusion gradient. Now, how does this affect the rate of diffusion? The higher the diffusion gradient, the higher the difference between high concentration and low concentration, the higher the rate of diffusion. So that if you have two regions where one side is very highly concentrated and the other one is lowly concentrated, then the rate of diffusion is faster, as opposed to where diffusion gradient is small. So the higher the diffusion gradient, the higher the rate of diffusion, and vice versa. The other thing also is temperature. Now, temperature affects in chemistry we do kinetic theory of matter. Now, kinetic theory of matter says matter is made up of small particles which are in constant random motion. These particles, when the temperature is increased, the vibration of particles increases. Now, when the vibration of particles increases, then the rate of diffusion also increases. So, when the temperatures are high, the vibration of particles is increased. I mean, the kinetic of energy of the particles increases. When the kinetic energy increases, vibration increases, then the rate of diffusion becomes more or increases. So the higher the temperatures, the more the vibration of the particles, and thus the higher the rate of diffusion. And the vice versa is also true. So that is it. Those are the factors that affect the rate of diffusion. The other one also we can include here is thickness of membrane. Thickness of membrane. The thinner the membrane, the higher the rate of diffusion. The thicker the membrane, the lower or the slower the rate of diffusion. When we look at the ileum, the small intestine, we say the ileum, the wall of the ileum, because diffusion of food takes place, absorption of food takes place in the small intestines. So in the small intestines, the lining of the small intestine is thin. We normally say it has a thin epithelial lining. Now that thin epithelial lining is for increment to increase the rate of diffusion. So the thinner the membrane, the higher the rate of diffusion. Now I want you to find out, this is your assignment now, find out application of diffusion. How is diffusion important in plants and roles of diffusion in plants and animals, in living organisms? So go find out that, and you can answer in the comment box. You can comment the uses of diffusion or the roles of diffusion in living organisms. So that is one of the physiological processes. Okay. Number 2 is osmosis. osmosis. Now I want to do this to summarize this very in a different dimension or in a different manner. Let me have two solutions here. Okay, let us have these two solutions here. This one, let me say this represents solute molecules. This one here, and this other represent solvent. Solvent molecules is water molecule. Solute molecules is any solid, can be flour, can be sugar, can be salt, anything. Now consider these two diagrams. What we can say about the first one is that we have more solvent molecules and less solute molecules. We have more solvent. The solvent molecules, these ones, are higher than the solute molecules. In this one is opposite. We have less solvent molecules and more solute. More solute, less solute. More solvent, less solvent. Now, a solution that contains more solvent molecules and less solute molecules is said to be a lowly concentrated solution. Okay? A solution that has more solvent and less solute is said to be lowly concentrated solution. A solution that has less solvent and more solute is said to be highly concentrated. Yes, a solution which has more solute molecules and less solvent molecules is said to be a highly concentrated solution. A solution that has more solvent molecules and less solute molecules is said to be lowly concentrated solution. And when you say high concentration, low concentration, now a lowly concentrated solution is said to be a dilute solution. Okay, a lowly concentrated solution, a solution which has more solvent molecules, is said to be a dilute solution. A solution which has okay, now let me complete this. So a dilute solution is said to be hypotonic. Okay, a solution which is dilute is said to be a hypotonic solution. A solution which is highly concentrated is said to be the concentration is high, is referred to as hypertonic. So this solution here is hypertonic. This solution here is hypotonic. Okay, now so a hypotonic solution contains more solvent, less solute. Hypertonic solution contains more solute, less solvent. I hope you are grasping that. Now, how do we define osmosis? Supposing this is this is A, this diagram A, and this diagram B. Osmosis now is the movement of solvent molecules from A to B through semi-permeable membrane. Now look at this. Osmosis is movement of solvent molecules from A to B. So you can say osmosis is the movement of solvent molecules from a region which contains more solvent to a region which contains less solvent molecules through the semi-permeable membrane. Okay, you can say osmosis is the movement of solvent molecules from a lowly concentrated solution to a highly concentrated solution through the semi-permeable membrane. Or you can say movement of solvent molecules through from a dilute solution to a highly concentrated solution through semi-permeable membrane. Lastly, movement of solvent molecules from hypotonic solution to hypertonic solution. So let me use the last definition. Osmosis, therefore, is the movement of solvent molecules from a hypotonic solution to hypertonic solution through the semi-permeable. Underline these words. Underline semi-permeable membrane and solvent molecules. Osmosis, diffusion, we just said it is the movement of, we have said diffusion is the movement of molecules. Okay, in short, actually diffusion is movement of solute molecules. Diffusion is movement of solute molecules from a region of high concentration, to a region of low concentration, just like that. That's diffusion. But now osmosis is a special kind of diffusion. Osmosis is a special kind of diffusion. Special in the sense that it is only involving the movement of solvent molecules from A to B. You can now use any of these terms to refer to A and B. But now you should have the following in your statement. You should have solvent molecules and you should have semi-permeable membrane. Without the semi-permeable membrane, there is no osmosis. Okay? Okay, so that is osmosis. So osmosis, movement of solvent molecules from, so you can say from solvent molecules from their region of high concentration to their region of low concentration. When you say their region of high, you mean where they are more, to their region of low, where they are less. Are we together? You don't just say movement of solvent molecules from high concentration to low concentration. You will have referred to from B to A. Okay, so that is the difference that we need to make. So for this fact, we now say osmosis is a special kind of diffusion. Now let me also bring in the issue of animal and plant cell in hypotonic and hypertonic solution. Okay, let me bring in and let me start with animal cell. Let me not take out this first because I'll be coming back here. So let us say animal cell I'll just bring it back. Animal cell in hypotonic solution. An animal cell in hypotonic solution. Now, hypotonic solution is A. Animal cell in hypotonic solution. So, when you take an animal cell and put it here, it means the animal cell itself is hyper. Okay? So, if you are taking an animal cell and putting it in an hypotonic solution, it means the animal cell is hyper. So from my analogy, this one is A, this one is B. Alright? Now, what happens? So an animal cell placed here. So what will happen? So the animal cell is B and the solution put in is A. So movement of solvent molecules from A to B. So now that means if we are putting it in hypotonic and it is hypertonic, so there is, it now takes in water. So the animal cell takes in water by osmosis. Okay. After it takes in water, it swells. Right? When you take an animal cell and put it in hypotonic solution, it will absorb water or it will take in water by the process of osmosis. When it takes in water, it will swell. Okay? Now, when it continues to, it swells until it bursts. So we now say the swelling and bursting of an animal cell, let's say a red blood cell, of an animal cell, of a red blood cell, when put in hypotonic solution, is called hemolysis. Hemolysis or hemolysis. That is in hypotonic solution. What about in hypertonic solution? Animal cell in hypertonic solution. So the opposite will happen. If you take an animal cell and put it here, so if you take animal cell and put it at A itself, it will be hypertonic. It will be highly concentrated. So it will take in water. The way B is taking in water. Taking in water is what we have said, movement of solvent molecules. So it's like B is taking in water. Now if it is put here, it will be now, the cell will be hypotonic. Okay? So if you are putting it in hypertonic in our discussion is B. So animal cell will be A. And we have said movement of water molecules from A to B. So if you are now putting it here, it means it will lose water. When it loses water, it shrinks. So it loses water and shrinks. Now this one, the shrinking of an animal cell, we can use the red blood cell still, of a red blood cell loses water by osmosis, if you like, by a red blood cell when put in a hypotonic solution is called crenation. It is called crenation. So the cell becomes crenated. So it becomes crenated. So the cell shrinks and becomes crenated. We can add that there, and becomes crenated. So that is that's what happens. Now the shrinking of the red blood cell as a result of losing water when put in hypotonic solution is said to be crenation. Okay, so so class, let that be our lesson for today. So today we have looked at physiological processes, two of them. We have looked at the membrane, cell membrane. We have said it is semi-permeable. It is a living membrane. We have discussed diffusion. I have told you to find out the role of diffusion in plants and animals. Now here we have discussed hemolysis and crenation. Those should be the terms of the day. We have crenation and hemolysis. You should be able to explain hemolysis. You should be able to explain crenation. So we can pick up from this in our next lesson. Otherwise, I want to say thank you for coming back. Thank you for tuning in. I know we were inconvenienced in the first place, but we are now okay. So in our next time, we shall complete the rest. We shall look at plant cell in hypotonic solution, and then we summarize with the active transport. So thank you for coming through. Keep checking, keep commenting, keep liking. YouTube, you can go there, subscribe, and God will bless you. Otherwise, until next time, bye bye.